Okay, thank you for asking me to present, and it's an honor to present here at DareFest. And I'm going to dovetail on Fred's presentation and tell you about understanding metabolic signatures using metabolomics, what we hope to do with Refsum disease by kind of explaining some of the things that have been done in the field on other peroxisomal disorders. My name is Michael Wangler. I'm trained as a pediatrician and medical geneticist, and I'm at Baylor College of Medicine. So metabolomics, as Fred had explained it, is a method that we've been using at Baylor College of Medicine. And the problems that we hope to address is how different peroxisomal disorders, of which Refsum disease is one, impact the total body chemistry. And we hope to understand the biochemical relationships between animal models, as Fred talked about the mouse, and disease states. And then one of our specific interests is to understand how different alterations in lipids can impact myelin in the brain and other disease states. So the idea behind taking a metabolomics approach is that we all have encoded in our genomes sort of genetic determinants, but those are in between our genetic determinants and the actual traits or, say, medical conditions that we have, that in between those stands our overall biochemistry. So a lot of our genome is actually geared towards making proteins that may be involved in biochemistry. And then that biochemistry can feed forward and sort of determine our phenotype. So we can see metabolomics as being a bridge between a person's genome and their phenotype. And the other advantage is that we can look at many different sample types. We can look across many different compounds. So it's a very information-rich approach. And it may be actionable more so than a lot of other genetic changes. So we may find particular compounds that we could add or take back that may actually help a medical condition. And so for all these reasons, we can kind of see that going from a person's genome that leads to the RNA to proteins, and then what we focus on in metabolomics is the small molecules, sort of in between what the proteins are doing and the insight into the phenotype. And so thinking about this another way, you can think about that our genomes have an array of common variation that's maybe passed from a larger population, whatever population we belong to. There may be very rare variation that's seen just in, like, say, our family or just in us. And then there's also environmental determinants like nutrition, exercise. And then these would feed into what we think of as intermediate phenotypes, what's happening with gene expression, what's happening with our proteins, and then what's happening with metabolites. And that could lead to different molecular states and could affect our ultimate susceptibility to disease. So one of our areas of focus here at Baylor has actually been looking at this and using metabolomics, and as well as genomic sequencing and animal models in a different set of peroxisomal disorders, the peroxisomal biogenesis disorders. I wanted to show you some of our data because we would really hope that some of the things that we've learned in these conditions may feed forward in further studies on Refsum disease. So in the peroxisomal biogenesis disorder study that we've been running at Baylor, we've been recruiting patients to our study, incorporating medical genetics evaluation, looking at specific sequence variants, and then we use metabolomics and animal models to try to understand more about mechanism, and then perhaps to learn better ways to get at molecular diagnosis in the future. So the platform that we're using has a lot of similarity to what Fred described, but involves a collaborative effort with a researcher here at Baylor named Sarah Elsey. So we basically, from a sample, extract, and then 4 different mass spec platforms are applied to that sample. Compounds that are detected are kind of filtered through a library search to compare them to known compounds. And then you get essentially a spreadsheet and an interpretation. And the hope is that this type of work will help us have biomarkers, mechanistic understanding. And we also hope that there may be drug targets that could be identified. So with the peroxisomal disorders, sort of 2 classes of biogenesis disorders, the Zellweger spectrum and the rhizomelic chondrodysplasia punctata, which I'll from now on refer to as RCDP, have been worked on with a number of samples undergoing metabolomics in our group. And you can see that we've actually published some results on both animal models and human subjects for these disorders in relatively small samples, but in enough to get a sense of some of the patterns. So this shows for the Zellweger spectrum disorders our findings. And the way that some of these metabolomic studies are done, you can make what's called a heat map. So what we have here is that red shows you a specific compound that's increased. Blue shows you a specific compound that's decreased. And so basically each column here is an individual's plasma sample. And then each row is a specific body chemical, a specific compound that was found in that sample. So in the Zellweger spectrum disorders, we actually learned that we could see increases in things like long chain dicarboxylic acids. And we became very interested in the sphingolipids, where we saw relative decreases. So what you can see is that every person is different. There's variation between individuals. So each column doesn't look exactly like each other column. But when you put individuals together in a group, you can identify specific patterns that may help you flag certain compounds. In this case, for us, sphingolipids is something of importance that we're studying in Zellweger spectrum. Another thing that I think was interesting is that we actually saw that things that were abnormal in the Zellweger spectrum were not abnormal across ages. So here, each point is an individual, and then here across the x-axis is shown how old they were at the time we collected the sample. So what you can see is that individuals who are younger actually had more significant changes. And then with age, a lot of the biochemical features seem to normalize. Kind of to show that another way, you can see that again, the red is decreased, the blue and purple represent increases. And so if we look at the individuals who are under 10, we can see a lot of different parts of our biochemical tree that all sort of light up with increases and decreases. But then when we look at the individuals who are over 10, there's a lot fewer changes in the overall metabolome. So this is another type of insight that we can get from metabolomics, is that it doesn't appear that the samples are the same over time. So an individual who's older, you might expect a different array of changes than an individual who is very young. And another part of our work is that we really hope to develop sort of a Venn diagram across many different peroxisomal disorders. So in this example, what I'm showing you is that the ZSD condition and the RCDP condition, 2 different types of peroxisomal disorders, that we can see that there's a defect in plasmalogens, a specific biochemical, in both of these samples. Now, one group has a much greater deviation, a much greater difference, but in both groups, this same analyte is altered and is low. On the other hand, we can see that there are specific compounds that are only altered in the RCDP group versus the ZSD group. So rather than, the specific compounds may or may not be of significant importance, but in a lot of metabolomic studies, we're looking for patterns that would help us distinguish one group of patients from another group of patients. And so this is sort of showing that there are changes that appear specific for one peroxisomal disorder, not seen in another disorder. And I mentioned that we were very interested in the sphingomyelins, and this was another example where we can see those seem to be very specific for the Zellweger spectrum, but not seen in the other peroxisomal disorders that we've looked at so far. So the metabolomics really gives us insight because not only can we look at many different compounds, if we look across many different groups of related disorders, we can start to build a picture of what characterizes and what's unique about each condition. So our hope in sort of being invited to DareFest 2022 is to sort of explain to you the use of metabolomics and hope that you get a sense of the promise that this sort of technology would have in the field. And we hope that with Fred and ourselves and other people in the field who are using this methodology, that we could do a more detailed study of Refsum disease and really understand what are the key metabolic changes that are seen. So with that, I would really like to thank you for inviting us to participate in DareFest 2022 and thank Christy in particular. And then these are I want to acknowledge the team and funding sources for the work that I presented to you related to RCDP, ZSD. So with that, thank you very much for your attention.